Swayam Prabha. Digital India. Educated India.
Welcome to the structural biology course again. Today it is the module protein which we are continuing. Today our discussion will be protein chemistry, chirality, peptide bonds and level of different protein structures. In the last class I talked about the chemistry, the position of the atoms in the periodic table, the involvement of hydrogen, carbon, nitrogen, oxygen as well as sulfur and phosphorus. We have looked at their electronic outer cells and we also have seen that how they develop sp3 hybridization to develop a tetrahedral structure. For carbon, it have the tetrahedral structure, it have the carbon orbital as rigid steric strain which makes the basic building unit of the protein conformation. So, what it was actually doing when we look at the hydride of a carbon, we see that it have the formation of a classic tetrahedral with the angles 109 degree 28 minutes. But in addition to this gives another interesting phenomena. If you look at the carbon in this model, you will see that this is the tetrahedral. Now, if you replace instead of hydrogen, if these are 4 different groups, then suppose this is carbon and they are group 1, group 2, group 3 and group 4. What will happen? A very interesting phenomena we discussed called chirality, where if you see, if you want to put this in a plane, you could only develop a plane with 3 of them, like these 3, the carbon group 4 and group 3, or the carbon group 2 and group 3, or the carbon group 1 and group 2, or group 1 and group 4. How that change the scenario? That change the scenario because if you want to put these 3, if you look at my hand, these 3, you will clearly see that one group would be above the plane and another group would be below the plane, and these we call chirality. And amino acids have chirality because it have a carbon, it have a CO2H group, it have a NH2 or NH3 group, it have a hydrogen, and 19 of the amino acids among 20 have a are different than hydrogen, only glycine have the hydrogen. So, we talked about the chirality. Amino acids except for glycine have a chiral carbon atom adjacent to the carboxyl and amine groups. This chiral center allows for stereoisomerism as there are 4 different chemical groups surrounding the carbon atom. The amino acids with 2 stereoisomers are mirror images to each other and are called L amino acid and D amino acid. So, if you see there, stereoisomers, they could not superimposed. In glycine both of them are hydrogen, in others they are different, the R groups. In alanine you see the CH3. So we know about those. We, as I told, the structures are not superimposable, but at the end what we have to understand, we understand that amino acid have a carbon, the carbon have 4 different groups, and that is why they are chiral. How or why this chirality is affecting is what you have to understand. So, if you see, there are 2 groups which are, one is above the plane and another is below the plane. When some ligand would be approaching this carbon, what will happen? It had to come on the side of the above the plane or below the plane. So, if you think it is a chemical reaction, the side it would take most probably which is having less steric. So, if we think that group 1 and group 2 in case of amino acids, group 2 is hydrogen. So, because of the chirality, there is a directionality in the enzymatic reaction. So, it would contribute to the reaction specificity, and I would follow this with a very interesting story which will help you understand the importance of chirality. Peptidoglycan or murin is a polymer consisting of sugars and amino acids that form a mesh-like layer, a network outside the plasma membrane of most bacteria, forming the major component of the cell wall. The sugar component consists of altering residues of beta 1,4 linked N-acetyl glucosamine, which is called NAG, and N-acetyl muramic acid, which is called NAM. So, this is the structure of N-acetyl glucosamine and this is the structure of N-acetyl muramic acid. They form linear chains which is known as peptidoglycan layers. In the cell wall, there are many such layers, but the layer would be strengthened that only when they would be cross-linked. So, the layers are cross-linked, NAM to NAG, but it could be opposite way too. And this cross-linking is done by a pentapeptide. They might vary, but mostly they are L-alanine, D-glutamine, L-lysine, D-alanine and D-alanine. I talked about that the amino acids could be D or L and they are normally having the L amino acids and when they cross link, it looks like this cross-linked network gives very good stability to the cell wall. So, what I want to tell here, if commonly the amino acids are in L, then why here you see different or opposite chirality? This is because in the cell there are proteases what protease do? They cut the bond between, which we are going to talk about, two amino acid which are called peptide bonds. But with the presence of opposite chirality, they are not supposed to cut it. So, nature understand the importance of this cross linking in the survival of bacteria, so they have incorporated opposite chirality so that the common proteases could not cut this. So, that is what I was talking about. The chirality is extremely important towards biochemical reactivities, enzymatic activities. There are enzymes which are specific to some conformers like L amino acids. There are some who are having broad specificity, and depending on those differences, a lot of different biochemical, biophysical phenomena are altered. We will see them. And when I am talking about these peptidoglycans, I would not leave the opportunity to introduce the mechanism of beta lactams. Beta lactams, if you guys do not know, is the mostly prescribed antibiotics. What I mean is, if today you go to a doctor and tell that you have headache, you have any other symptoms which apparently needs treatment, the first treatment or first prescription doctor made, if you get 3 medicines, 2 of them must be beta lactams or beta lactam based beta lactamase inhibitors. So, what is beta lactam? Beta lactams are antibiotics. Why they are so important? I already talked about the peptidoglycan layer. You see there is DLA cross linking, and when you look at the DLA moiety, this cross linking is done by enzyme called transpeptidase. Now, if you look at penicillin, which is the first beta lactam, you will see that it had remarkable similarity with the DLA. Because of that, penicillin or other beta lactams, which I will talk later, sit down in the active site of the transpeptidase. That is not the end. Here you see a 4-member beta lactam ring. If you do a simple mathematics, you understand what I am talking now. If you look at biology, you look at DNA, you will see 6-membered rings. You will see 5-membered rings. Same you will see in sugars, in proteins. Why biology prefer 5 or 6-membered rings? Because the strain generated because of the angular changes is least here. And the strain is most in a 4-membered ring or a 3-membered ring. Because of ring strain, they are very reactive. So, when they sit down at the active site of the transpeptidase, they react and ring breakage happens. Because of that, remember, a very, very unique phenomena of biochemistry I am talking about. I am talking about reaction between an enzyme and a substrate. If you guys remember the definition of enzyme, you will definitely recall every time we talk about enzyme, we say that enzyme don't take part in the reaction, and this is a very good example where the enzyme itself take part in the reaction, form a covalent bond. As a result, the enzyme transpeptidase become inactive because it form a covalent bond with the substrate. So, it could not further take part in the cross linking of the peptidoglycan layer. So, what happened? Now, the peptidoglycan is not cross linked. So, the cell membrane of the bacteria become weak because they do not have the cross linked peptidoglycan. So, with the exposure to hypotonic environment, the membrane will be bulges out as water will be diffuses, membrane breaks and ultimately resulting the death of bacteria. So, that is the reason beta-lactam antibiotics are extremely popular. When I talked about beta-lactams, I will finish by talking about the story of penicillin. You all know about Fleming and his experiment. He was working on bacteria S. aureus, and the picture you are looking here is a petri plate where he grow the bacteria. When bacteria grow, it become visible, that is called colony. So, these are normal bacterial colony. Now, he was working on them and he went to vacation keeping the plates there. When he came back, he found that in one part of the petri plate there is penicillium colony. What makes him excited? Around the penicillium colony, if you see here in the picture, the normal bacterial colonies were not existent. That gives him an idea that might be this penicillium colony is giving something or doing something so that the bacteria are killed. So, he picked up that penicillium colony, applied it to different bacterial stocks and confirmed that yes, this penicillium colony have antibacterial activity. In his own language, because it is exciting, when I woke up just after dawn on September 28, 1928, I certainly did not plan to revolutionize all medicine by discovering the world's first antibiotic or bacteria killer, but I guess that was exactly what I did. Now, this is protein module. I do not go further because I will talk about penicillin and beta-lactams in different section. Just to finish up, because of his contribution, Fleming was awarded Nobel Prize in Physiology in the year 1945. Rest of the things we will talk about: story of penicillin, story of beta-lactam, story of beta-lactam maize, story of antibiotic resistance is what that is critical for existence of ours. COVID have already shown how a small virus could do, and trust me, there are many bacteria like that who could have attacked us, kill us, even with more potent strategies than what COVID is taking now. We started talking about the levels of protein structure. primary structure, secondary structure, tertiary structure and quaternary structure. So, from primary structure, like how the primary structure form is come through the formation of something called peptide bond. We have amino acid 1, amino acid 2, then there is hydrolysis and formation of peptide bond. So, the C double bond OH NH3, they combine, they leave a water molecule and connected. So, a peptide bond is a covalent linkage between two amino acids formed by the condensation reaction. This bond is formed between the amino and carboxyl groups of two successive amino acids by hydrolysis of one water molecule. How the peptide bond form? In the first step, two amino acids are linked together. Here the acid group of the first amino acid should be close to the amine group of the other amino acid. Step 2, the water molecule is eliminated, leaving a bond between the acid carbon of the first amino acid and the amine nitrogen of the second amino acid. Step 3, the peptide bond is therefore formed between the two amino acids. So, we talked about in the last class that how the presence of those fundamental elements hydrogen, carbon, nitrogen and oxygen have developed biology. We showed you the functional groups and I talked about two things. One is oxygen, which is providing us negative charge, and we showed that oxygen is mostly electronegative with 3.5 in the scale of electronegativity. But very interestingly, there is nitrogen which actually comes up with electronegativity 3. So, it definitely could provide us negative charge, but in addition it form NH3 plus. So, it also provide us positive charge. Remember, I talked about that now we have the polymers, and if a polymer form because of the catenation property of carbon, it develop a backbone which is neutral, but what we need is the spicing up, and that spicing up is making by the introduction of negative charge by oxygen and nitrogen and positive charge by nitrogen. In the last class, we have also discussed a very important point. I want to reiterate that when we are searching for positive charge and you are looking biology, there are metals which could have provide us positive charge, and it is not that metals are not involved in biology. There are Mg2 which is very illustriously present in biology. There are iron, there is copper. So, metals are being present in biology, but we cannot incorporate that directly by developing a chemical bond. We want much milder chemistry in a polymeric composition, and that is the reason the entire polymer formation in biology have done with carbon followed by hydrogen. Hydrogen always fill up the space. So, carbon is neutral, hydrogen is neutral, and then nitrogen making positive and negative and oxygen giving negative charge. We also talked about bond length calculation. I have shown in the last class how we have taken the bond lengths of carbon single bond, carbon double bond, carbon resonating bond, oxygen single bond and double bond, nitrogen single and double bond, hydrogen single bond and sulfur single bond. We have shown the bond lengths and we told that when a bond is formed, we do not have to do much, we have to just add the bond lengths. Here I have given the bond length of the peptide bond and you could just check how we are getting the additions. When you have two carbons, you add the single bonds. When you have double bond, you add it, and you keep getting the idea how this bond length calculation made. Now coming to another aspect. So we have carbon. We talked about carbon is having 2s2, 2p2 in its ground state. In its excited state, it form 4 sp3 orbitals and they do the formation of the sigma bonds. But now we will consider the pi bond. So this is the sigma bond between carbon and oxygen, where they have the overlapping. Now the pi bond would be formed where they will use the pi electrons to develop the bond, and sigma bond, because the overlap is straightforward, is having higher strength, whereas pi bond would have a relatively lower strength. Now the situation comes like this, but here it have a lone pair, and that lone pair would be used to form a double bond. So now it would form a situation like this, where if you see now they have a delocalized state where there would be resonances. So this is one, this is the another one. Now because of that you see a partial double bond in between carbon and nitrogen. So I am showing you a peptide bond. This is a peptide bond, and you see that in the peptide bond it's heavy. So, this is a peptide bond, and if it is single bond, there would be free rotation, but because it's partial double bond, so that rotation is restricted. Also, how it is restricted? It is restricted depending on the size of the amino acid side chain. So, what is apparent here, that one, the partial double bond would not allow free rotation to the restriction would be further implemented depending on size of amino acid side chain. These two are very, very important factors, and these, as I told sometime, make protein different from other polymers. Why? Because in other polymers, if you imagine, be it carbohydrate, be it DNA, be it RNA, all the connections are made through single bond. Now here in the junction we are getting a double bond, so there are restrictions, and again with different size of the amino acid the restrictions are different, gives us a chance to model, to control. What I mean? I mean, if you have a carbohydrate, it could have any conformation, so you cannot make any rule. But because here we have the partial double bond along with the difference in size, along with the existence of only L amino acids for the 19 chiral amino acids, you could dream of controlling, you could dream of modeling, you could dream of having the rules, and that is what happening in case of proteins. So, peptide bond, the structure of the protein is partially dictated by the properties of the peptide bond. You see the contributions, I talk about two, there is a third one which is minor contributed. The peptide bond is a resonance hybrid of the canonical structures, which you could see here. The resonance causes the peptide bonds to be less reactive compared to esters, to be quite rigid and nearly planar, to exhibit a large dipole moment in the favored trans configuration. So, it is double bond, there is a possibility of, when you have a double bond, you have a possibility of cis trans. You have to remember, in case of protein amino acid, this is hydrogen, sorry. So, these are the side chains. So, side chains of first amino acid and second amino acid in a peptide bond would be returning in a trans configuration. This brings us to secondary structures. The secondary structure refers to local spatial arrangement of the polypeptide backbone. There are two regular arrangements are common. One is alpha helix, which is stabilized by hydrogen bonds between nearby residues. The beta sheets, stabilized by hydrogen bonds between adjacent segments that may not be nearby. Irregular arrangement of polypeptide chain is called random coil, loop or turn. So, there is alpha helix, there is beta sheet, and there are irregular ones which are called either loop or turn or random coil. Alpha helix is a common motif in the secondary structure of protein. It is a right hand helix conformation in which every backbone NH group hydrogen bonds to the backbone C double bond O group of the amino acid. So, if you remember, when you have a peptide bond, you have this. So, this is the CO, the CO hydrogen bonds to NH, definitely not the adjacent. In case of alpha helix, it is N to N plus fourth residue. So, it is a right hand helix conformation on which every backbone NH group hydrogen bonds to the backbone C double bond O group of the amino acids. So, if you see here, always it starts from the amino terminus. Remember when I was showing you, it starts from the amino terminus and it goes to the carboxy terminus. It is always like that. You will see the pitches which I am talking about. Very interestingly, you could do that experiment by your own, how alpha helix is the most stable structure. You start thinking, I will answer you in the next few slides. So, alpha helix was first proposed by Linus Pauling and Robert Corey in 1951. Identified in keratin by Max Peruz, a ubiquitous component of proteins, and stabilized by hydrogen bonds. Simple. The alpha helix is actually a coil and is the most stabilized component of protein. As I told, it shows the effect of gravity on us. What I mean by that? What I mean is, if you guys could take a drop of water and you allow the water in the top of your finger and allow that to drop and watch carefully, you will see that the water becomes spherical. That is the effect of gravity, and because of that, every entity for their stability want to, especially polymers, want to shape spherically. The apparent spherical tendency of a polymer allow them to become helix. If you do not understand what I mean, when we will go to the visualization section, you see here the alpha helical molecule, you take it and you turn it in such a way that it would come from this point to this point like this in front of your eyes. You will see that you get a circular structure. So, and you also, if you think, in case of DNA also you get a helical structure. So, all the biological polymer have a tendency. How the residues stay there? Per turn there are 3.6 residues and rise per residue is 1.5 angstrom, which provides rise per turn, that is called pitch, 3.6 into 1.5 angstrom, that is 5.4 angstrom. The amino hydrogen bonds with carbonyl oxygen located 4 amino acids away forms 13 atom loop. The average length 12 amino acid is 3 turn and approximately 18 angstrom length. The R group, the side chains point outward, generally downwards. If you look at alpha helices, they have a directional motif. So, all the hydrogen bonds in the alpha helix are oriented in the same direction, giving a helix a dipole with the N terminus being positive and C terminus being negative. So, if you see here, in the N terminus it is plus 2, in the O it is minus 0.42. Now, the helix dipole result from the orientation of the CONH hydrogen bonds. You see, you get delta plus in the amino terminus, delta minus in the carboxy terminus. What does this imply for the preferred placement of positive and negative amino acids along alpha helix? So, you come to the wheel and you see that, as I told, now you see that it looks like a sphere, and you could see that have implication. What type of implication? If you put them together like I have put it here, you see that the hydrophobic residues comes in one direction of the wheel and the other residues, including the hydrophilic, comes in the other side. Why these are important and what is the implication? This will tell you how one helix will interact. This will tell you what residues should be kept in one position if you are doing enzyme engineering, protein engineering. We will see them in later sections. But the way the helix is developed, definitely there are rules. More you understand the rules, better you understand the protein. Side chain groups, as I told, point outwards from the helix. Amino acids with bulky side chains, less common in alpha helix. Glycine and proline destabilizes alpha helix. This I talked yesterday, last class. Glycine being very small, it brings flexibility, and proline, if you remember the locking, they force to make a turn. Amphipathic alpha helix. So, one side of the helix has mostly hydrophobic amino acids. Two amphipathic helices can associate through hydrophobic interactions. Here you see the example which is called leucine zipper. Leucine zipper is a motif, super secondary structures which we will talk later. Here there is a helix interaction stabilizing the motif, and it could bind to DNA. So, that's once you see a leucine zipper, you know that it is binding to DNA. It's a DNA binding motif. We will talk about that later. So, in summary, most common type of secondary structural element, about 25% of the amino acid in protein are in this structure. Right-handed helix. R groups project outward and provide the main constraints on the helical structure. Stability is greatly enhanced by internal van der Waals contacts. Hydrogen bonds are in line optimum distance and having amphipathic properties. There are commonly three types of helices. We talk about alpha helix, we talked about amino acid per turn 3.6 and the frequency of existence is 97%. There are other ones, 310 helix and pi helix. 310 helix, amino acid per turn is 3 and existence is 3%, and pi helix amino acid per turn is 4.4 and existence is rare. So, there are structures having alpha helical like vimentin, which is a bundle of alpha helices coil motif. So, alpha helices are about 10 residues in average. Side chains are well staggered, preventing steric hindrance. Helices can form bundles, coils, as this is an example. Now, we are going to talk about beta sheet and beta strands. Individually, it is called beta strand, but you understand, if you look at a beta strand, it cannot form hydrogen bond with itself like an alpha helix. So, you cannot calculate the stability or energy of a beta strand. What you have to do is to consider a beta sheet which is formed by multiple beta strand and they have hydrogen bond between each other. It is not individualistic, it is a hydrogen bond between one beta strand with other beta strand, forming a beta sheet. It was also first postulated by Pauling and Corey in 1951. They, as I told, may be parallel or anti-parallel. The rise per residue is 3.4 angstrom for the anti-parallel strands and 3.25 angstrom per parallel strands. We are going to discuss them later. Each strand of a beta sheet may be pictured as a helix with 2 residues per turn. So, beta sheets are highly extended form of polypeptide chain, 3.5 angstrom between adjacent residues, which is 1.5 angstrom for alpha helix. Adjacent side chains point in opposite directions. A beta strand usually is associated with another beta strands. So, when they are anti-parallel, they form a structure like that. They are stabilized by their hydrogen bonds, which are head to head. Generally, the R group orientation maintains certain distance, it have a pleated character and average strand length is about 6 amino acids. So, when we look here between 2 nitrogen atom, the distance and how they are coming here between one point to the next is 0.7 nanometer, which is 7 angstrom. Parallel hydrogen bonds not at optimal angle, because if you see they are having an angle, they are not head to head. The R group orientation is also different and they also have pleated character and average strand length is 6 amino acid. And the distance, as I told, between the two turns, it is reduced from 0.7 nanometer to 0.65 nanometer, 7 angstrom to 6.5 angstrom. Coming to loops and turns, loops usually contain hydrophilic residues found on surfaces of the proteins, connect alpha helices and beta sheets. Turns, loops with less than 5 amino acids are called turns. Beta turns are common. What are beta turns? Beta turns occur frequently whenever strands in beta sheets change the direction. The 180-degree turn is accomplished over 4 amino acid. The turn is stabilized by a hydrogen bond form a carbonyl oxygen to amide proton, 3 residues down the sequence. So, it is similar like alpha helix, but instead of n to n plus 4 it is 3 residues. Proline in position 2 or glycine in position 3 are common. So, there are 2 type of beta turn. In the type 1 beta turn, in the second position there is presence of proline. In the type 2 beta turn, in the third position there is presence of glycine, and this is generally the hydrogen bond formed in the beta turn. Also about proline isomers which we talked about. Most peptide bonds not involving proline are in trans configuration. For peptide bonds involving proline, about 6% are in the 6 configuration. Most of these are involving beta turns. Proline isomerization is catalyzed by the enzyme proline isomerases. So, this is the 2 conformation turns and cis conformation of proline. Coming to next level, protein tertiary structure. The tertiary structure refers to the overall spatial arrangement of atoms in a protein, stabilized by numerous weak interactions between amino acid side chains. Largely hydrophobic and polar interactions can be stabilized by disulfide bond. So, as I told initially, the covalent bonds have done their work. Now, starting from the secondary structure to tertiary structure to quaternary structure, it is the dominance of the non-covalent bonds which we will specially study after this section. We will see how they are working, what are their nature and how they influence, but mostly we could say that the hydrogen bonds, salt bridges, hydrophobic patches, base-base stacking, they have immense role in protein folding. Interacting amino acids are not necessarily next to each other in the primary sequence. 2 major classes of protein, fibrous and globular proteins. Mostly fibrous proteins are structural protein and globular proteins are proteins which work as an enzyme. Quaternary structures, multi subunit proteins have another level of structural organization known as quaternary structures. So, if your protein is only one subunit, it is the sequence became alpha helical, beta sheets and all, and then it goes to secondary, tertiary. Now, if they have multi subunit, they are coming together forming oligomers, dimer, trimer, tetramer and many others could be possible. A quaternary structure is formed by the assembly of individual polypeptides into a larger functional cluster. Quaternary structure refers to the number of subunits, their relative positions and contacts between the individual monomers. So, as I told, there are 2 type of proteins, fibrous and globular protein. Fibrous protein, they are made up of polypeptide chains that are elongated and fibrous in nature or have a sheet like structure. These fibers and sheets are mechanically strong and are water insoluble. They are often structural proteins that provide strength and protection to cells and tissues. Examples are collagen, fibrin, keratin. Collagen, it is the most abundant protein in vertebrates. Collagen fibers are a major portion of the tendons, bone and skin. As you see here in the tendon, bone and skin, they have collagen. Alpha helices of collagen make up a triple helix structure, giving it tough and flexible properties. So, this is the first chain which comes together and make a triple chain, and that is the final collagen structure. Fibrin fibers make the silk spun by spiders and silkworms stronger weight for weight than steel. So, you all have seen how the spiders are developing their nets. These are majorly fibrin protein. The soft and flexible properties come from the beta structure. So, this is the molecular structure where you see presence of glycine side chain, presence of alanine side chains. They are actually giving the structural rigidity. Keratin, keratin is a tough insoluble protein that makes up the quills of ecdina, hairs, nails, feathers, horns and the rattle of the rattlesnake. The structure comes from alpha helices that are cross linked by disulfide bond. So, these are the examples, snails, and this is the keratin structure, the alpha helical structure. How the structure organizes, we will talk in details with the molecular structure. So, this is just an example here. The globular proteins have a number of biologically important roles. They include cell motility, proteins linked together to form filaments which make movement possible, organic catalyst in biochemical reactions which are called enzymes, regulatory proteins, hormones, transcription factors, etc. Membrane proteins, MHC markers, protein channels, gap junctions, defense against pathogens, poisons, toxins, antibodies, complements, transport and storage, hemoglobin and myosin. So, here you see phosphoribosyl transferase which is an enzyme, IGF-2 a hormone, monoclonal antibody which is antibody giving us defense, hemoglobin is for blood transport, some other globular proteins, myoglobin, it is a monomeric protein that has 154 amino acid residues. It consists of 8 alpha helices connected through the turns with an oxygen binding site. So, we talked about hemoglobin. Hemoglobin is a multimer protein, myoglobin is a monomer protein. Myoglobin is the one of the first protein which was studied and crystallized. Triose phosphate isomerase, this is called TIM. It is a homodimeric protein, a 3D structure of subunit. It is a beautiful structure. It has 8 parallel beta strands surrounded by 8 alpha strands. TIM barrel proteins are like available in many domain of the life. Triose phosphate isomer is the first protein where TIM barrel was actually found. The first structure was solved because this is part of our most abundant metabolic pathway which is glycolysis. 20S proteasome, it is a complex of 28 subunits. It is a well organized protein complex with a sedimentation coefficient of 20S. The protein complex appears as a cylinder like structure. So, these are 3 structures we have talked about them. One is monomer, one is dimer and another is multimer. So, in that way organizational complexity enhances and protein from one protein structure it becomes molecular machine. We will study about them. So, today we have studied about chirality and their dependence, the chemistry behind the peptide bond, the secondary structural components, alpha helices, beta sheets, and ultimately we talk about few proteins, especially a special example of fibrous and globular proteins, and we have shown that fibrous protein which are mostly involved in developing structure, whereas the globular proteins are doing functions. One of the important function is definitely as enzyme, but there are other ones like transport, other one like defense and whatnot. In the next section we will go even deeper, talking about the angles which is there around the peptide bond and how they influence us to understand and predict the future structures. Thank you very much.